an environment that has a high level of degradation, and I'll talk about that in just a minute. And as I mentioned, there are many copies, as you see up in the cell, many copies of mitochondria, and so many copies of that particular gene. So two nuclear versus thousands of mitochondria. And for most organisms, the CO1 gene, the CO1 barcoding gene, is known for a variety of different organisms. And so oftentimes, when we're using environmental DNA, we capitalize on the CO1 mitochondrial gene. And the story I'm going to tell you about today with the fish markers is not so great for that, because we're not using CO1, we're using 12S, which is another mitochondrial marker. But nonetheless, a lot of mitochondrial barcodes are known for many organisms. So CO1 for most, and then 12S for fish. And one key thing that we do with environmental DNA is that we target short fragments. So because the DNA does degrade relatively quickly in an environmental habitat, we need to target like less than 200 base pairs. And so we're really using only about 150 to 200 base pairs to try to find the particular species or target goal. So other advantages to environmental DNA to make this tool sort of a great way to complement other methods is that it is can be efficient and non-invasive. So you don't actually have to have contact with your target species, and that's a thing that can be advantageous when the species is endangered or threatened or invasive or hard to find, or just elusive. And so you don't have to have that direct contact, and it's non-invasive into the habitat because you're just collecting water samples from the environment. So as I mentioned, it can be useful for those species that are hard to find, or when a conventional method, like using traps or balls or nets, might be damaging to the habitat or damaging to the organism itself. And then it's also advantageous, as you'll see in our study, for looking for small-bodied cryptic species. And so, when we're looking at a reef, which is where I'll go today in the Gulf of Mexico, there's the conspicuous players, the things that you always see that are there. But what about the things that just swam by? What about the things that are hiding down in the cracks and the crevices or the rocks of the reef? And those benthic species become difficult to sample kind of with conventional methods. So really what I'll argue for today is to use a combined method, to use environmental DNA and a conventional method, and to use those two together and we get this powerful look at the bigger picture. And I'm just going to show you environmental DNA data to get today, but the bigger part of the picture we're doing is using multiple methods to get a look at reef fishes in the Gulf. And then, as I mentioned, no direct contact is required with that organism. And in theory, there's no gear bias. So one of the problems that some of my colleagues have run into why we want to supplement with environmental DNA is because they use cameras to look at reef fish and to study reef fishes. And cameras have bias, right? So you put the camera down and it might scare people away, or it might draw other fish to it. Or what just swam by 3 minutes ago before you put the camera down, you didn't see that species or group of fish. And so there's less potential gear bias that might be introduced with different methods. Okay, and so overall, EDNA, I would say, is kind of reforming the study of marine diversity right now, or marine biodiversity. This is a great way to help catalog things, and we're finding things that were missed, or that are elusive, or we thought were extirpated that are actually there, or have invaded new habitats, as is the case with the lionfish. There are some things to kind of note about environmental DNA before we go any further, and one of those is that there are many things in the environment that do affect the persistence of DNA and how long it might stay in that particular habitat. And these factors are up on the slide here. Some of them are light, obviously UV radiation degrades DNA. Temperature, the higher the temperature, the faster the degradation. And then microbes and enzymes are really abundant in the ocean, definitely breaking down cellular debris. And so what this can help us infer is that on a short time scale, we know that that organism was there relatively recently. So we know that on the scale of a few days to a week in the ocean, that those species were just there in that particular habitat. And in the Gulf of Mexico, we do have a lot of stratification, and that does tend to work well for us when we're studying some of these reefs, because the surface, there's not a lot of vertical mix on a lot of our. Okay, and then another part that I want to introduce to you today is about metabarcoding. So we're not only using this DNA trace in the water column, but we're actually using a multi-species approach to try to identify who's there. So with this, we can look for multiple species at the same time, not just targeting one particular species. And what we use for this is a generalist marker and a universal primer. And so CO1 is a great generalist marker that is inventoried for many organisms on our planet. But it turns out that's not actually a great marker to use for fish. And so we'll talk about the marker that we're using for fish instead. But you can think about metabarcoding as more for less. So instead of just inventorying one species that you want to look for, say, the Alabama sturgeon, which is the project that we did, we could instead just try to look for any diversities that there. And then if we happen upon that, we check it off of the list in terms of that particular target species. So you get all of the species at one time, with some exceptions, but you get a list, a general list of who's there. And this was kind of really put forth for fish communities in 2017 by a study by Yamamoto et al. And their results were astounding. So basically, they did 14 years of visual surveys where they went every 2 weeks and visual surveyed this bay. And in 6 hours, they found that same amount of species with environmental DNA. So 14 years compared to 6 hours. Now, it's not always that simple. But that just shows you the amount of effort that can be put into a project in terms of person time compared to just a little bit of molecular time, the amount of species that you can still catalog on that same list. So that kind of revolutionized the idea when thinking about metabarcoding with environmental DNA, and then it has just taken off from there. Oh, okay. Should I stand here? Yeah. No, it's fine. Yeah. Can they hear me better here? Yeah, I'm a wanderer. Sorry, I'll stand right here. Yeah. Is that better? Okay. Okay, cool. That's okay. So for eDNA metabarcoding in fishes, we have to use the 12S RNA mitochondrial gene. And the reason for this is that CO1 isn't variable enough. So when we're looking at a primer or a gene that would be good for this, we want one that has a hypervariable region in the middle, but kind of universal regions on the side with not a lot of variability so that we can amplify that. And CO1 is not a good candidate for that. So actually 12S is. And these were primers that were developed by Maya et al. In 2015. And then they revamped them in 2020 and said, here's an even better set. But this is kind of the standard that's being used across the globe right now for eDNA metabarcoding of fishes. And as I mentioned, it amplifies a hypervariable region of the gene, and then we do bioinformatic processing, and then ideally, we end up with operational taxonomic units of who the species are from that particular habitat. And then we can inventory things based on family, genus, and species, and we identify those based on using BLAST. So the NCBI database or BOLD database where fishes barcodes are already placed. We then blast all of our results against that and try to get who's out there. Here's a look at that particular region in fishes. This is from Maya et al. 2020. And the basic takeaway from this is to look at each side. So you see here, everything looks the same. Here, everything looks the same. And in the middle, everything looks different. And so what our goal is to find a region where everything looks the same, but then gets different so we can tell who species is. And then everything looks the same so we can finish off that amplification. And so this is an ideal region for that, for doing eDNA metabarcoding in fishes. Okay, so I've given you the background now on eDNA and metabarcoding. And now I'm going to tell you kind of two stories where we've used these techniques. And the first is a little bit longer because it's a bigger project, but I want to kind of show you the utility and other things beyond the Gulf of Mexico and our water cycle. So the first story will be about reef fishes in the Gulf, and the second story will be about least terns. Okay, as many of you know, reefs in the Gulf of Mexico look very different from coral reefs, but there's still a major economic importance here in the Gulf. A lot of people live around reefs. They have lots of ecological services in terms of fishing, coastal protection, food, tourism, treatment for disease. So that extends like coral reefs. And we have a kind of a mixture here in the Gulf of Mexico. Reef habitats look different than like the Keys, but we still do have quite a bit of coral like animals on these reefs. And then, of course, fishing is a huge component of that, and particularly in the Gulf of Mexico, that's a huge commercial and tourist draw here for human dependence. And our reef fishes in the Gulf have major important ecological roles. Some of them are piscivorous fishes, some of them are corallivorous fishes, some of them are invertebrates, and so on, and they each fill this important ecological niche. Now we have artificial reefs and we have natural reefs in the Gulf, and the focus of this study was all on hard-bottom natural reefs. So there were no artificial reefs that were included in this particular study. And of course, we have commercially and ecologically important species. One of those is red snapper, which is a huge, focus study in the Gulf of Mexico, and now greater amberjack, where we're focused on the great snapper count and then the great amberjack count and so on. And despite all of this information about reef fishes and kind of what we know in the Gulf, we actually don't understand the assemblages super well on reefs. We don't have a good understanding of who's actually there. And the way that we study that is with camera data. So we put cameras down and we look at who's there and we watch the camera footage for a certain period of time, and we try to inventory, abundance and individuals and so on. But despite that, we still don't have a good understanding of who's on the reefs. Another thing that I want to mention about reefs before we get into our actual study objectives are the differences in complexity of our reefs here in the Gulf. So this can be kind of what a typical reef might look like. So you see a lot of soft corals and sponges and some kind of rocky areas. And it's probably not surprising that we do see a positive correlation between habitat complexity and species diversity, right? The more complex the habitat, the more species that we see. And when we're thinking about that structural complexity, so the differences or that 3D structure, that rugosity of the reef, is one of the strongest predictors of reef fish abundance, species richness, and biomass. Again, that's not shocking, kind of a general principle when we think about a reef and fish structure. So there's two major things that we are thinking about when we're categorizing our reefs based on complexity. And one of those is structural complexity, and the other is biotic complexity. So the first, structural complexity, is basically the 3D structure of the reef, or the rugosity that you see here. So we're looking at reef relief. So how high does the reef come up into the ecosystem? And that allows us to think about epifauna, or things that live on the reef as well. And then the other component that we think about with reef complexity is biotic complexity. And that's the coverage of microhabitats on the reef. So how much seagrass do we have? How much sponge habitat do we have? How much coral habitat do we have? Or rocky carbonate bank and so on. And that's that biotic complexity. Okay, so a little bit of background now on our techniques and on the reefs. And now I'm going to tell you the actual project objectives. So what we aim to do for this project was utilize environmental DNA metabarcoding to characterize species richness of fish communities according to habitat, and that's according to reef complexity in the Gulf of Mexico. And particularly, we wanted to examine how reef fish communities differ by reef relief, habitat complexity, depth, and then zone. And I'll tell you about the sampling in terms of zone. We collected at the surface water, and then we collected at the bottom water. So at the bottom of the camera, when it sits on the bottom, we then collected that water as well. So in terms of our methods, first we take a water sample. So we actually get an environmental sample. Then we extract the DNA from that. We then PCR it in the lab using those universal primers that I mentioned, those MyFish, those 12S primers. We get the sequence data back, and it goes through bioinformatics processing. And then ideally, we get taxonomic assignment at the end, and we can get a good understanding of who these fish are. And then, of course, we just correlate those data back to where they were collected, and the complexity of the reef, and so on. Okay, so for sample collection, these samples have occurred for the last 3 years, but I'm going to focus on our 2021 year of collections. That was last year. And this study is ongoing for the next, hopefully, 5 to 10 years as well. We have 2 more years of funding, and then we're up for renewal after that. So we collected them on the NOAA SRFB survey. So it's on the NOAA Pisces. And this ship goes out. We send grad students out with them all the time. And they're out there collecting our environmental DNA sample for us. These sites are predetermined and randomized each year, and we have sentinel sites that we're carrying through from year to year, but they're predetermined. So we don't get a lot of say in where these sites are going to be collected, but we know that they're all in the reef, and we do have complexity data for that particular reef. And we're using a method that is called the precipitation method. And so I'll talk a little. I'll talk a little bit more about that, just so you can kind of get an understanding of that. Conventionally, with eDNA, oftentimes we filter water. So we'll collect 2 liters, 5 liters, and then we filter that water. That's a great way to get a lot of water through a filter and hopefully get a lot of DNA from a water sample. Unfortunately, this method doesn't transfer across a variety of different ships, a variety of different people, and we literally have over 600 sites in the Gulf of Mexico, and that makes that incredibly onerous, high intensive, and then high risk for contamination. And so what we've done for this project is switch to something called the precipitation method, where we actually just take water, we pour it into a vial with sodium acetate, and we fill the rest with ethanol. And so we're collecting the water and we're not filtering it, and then it goes into direct extraction from there. And this method was established, like this is told, in 2008, and I've used it a bunch in the lab for several other projects. And as I'll tell you today, it seems to be going somewhat okay for this particular project. Okay, so we extract the DNA kind of in a standard way. We use the DNA blood and tissue kit. We then put it through PCR and try to amplify this 12S hypervariable region amongst those conserved regions using those MyFish U primers. And of course, along the way, we run extraction controls, we run PCR controls, and sequencing controls to help ensure that we are not getting any contamination along the way. And then after that, we'll run it through an agarose gel just to visualize our bands. And then, of course, we send it off for sequencing. And then when we get the data back, so we send it off for Illumina high speed. And then when we get the data back, it goes through a series of different bioinformatics processing. So for this particular part, okay, so we sent the products to University of New Hampshire. They have a core facility there where they do high-seq and high-throughput sequencing. And then after we get the data back, we use QIIME2. And QIIME2 is like a really popular piece of software to use for metabarcoding analysis and sequence analysis. And so we use QIIME2 on the outputs, and here we do demultiplexing, we do denoising, which helps get rid of low quality reads and things like that. And then we cluster the reads. Then after that, we get our operational taxonomic units. So we get our sequences back, and then we blast those in the NCBI database to hopefully end up with a match. This is an NCBI database. If you've ever blasted something, you're familiar with this kind of a image here. And then you can see we get an assignment for a particular taxon. Hopefully the species is identified. And for us to be an actual identified species, it had to match at 90% or greater, right? So if it was less than 90%, we said it was not a species match. Although we did include a lot of those data to genus level or family level or so on, but it had to be at 90% greater for it to be able to match to that particular species. Okay, so once we had the sequence data, we then wanted to go back and compare the sequence data to the environmental variables that we were interested in. First one we were interested in was sample zones. So were there differences between the surface samples and the bottom water samples that we collected on the reef? And then we wanted to look at sample depth. So did that matter if the sample was deeper? Do we see differences in those zones? And then of course we looked at reef relief and reef complexity. So just to give you a little bit of a background on how we analyze that in terms of complexity. So we have these pictures of the reef. So where we collected a water sample, there's also the camera data for that exact site as well at the same time. And it takes pictures of the reef and the fish that are on that reef. And it's analyzed with a visual ranking method, and it's categorized on a scale from 1 to 5. So 1 being low complexity, 5 being high complexity. And so in this picture here, this is an example of a reef that has biotic complexity of 4. And you can see here a variety of different microhabitats. So we see 5+ different what we call microhabitats throughout 80 to 20% of the scene. And so that makes this reef a biotic complexity of 4. And this is based on a method from a colleague of mine who's on the NOAA Restore project named Matt Campbell, who's over in Pascagoula at the NOAA office there. And then another example of a lower complexity, so this is a reef that has biotic complexity of 2, where we see only 3 to 4 microhabitats, but the coverage is less than 20%, right? So you do still see some soft corals and some things in here and some rocky habitat, but it is much less compared to that first image. So that's kind of how we score these different reefs based on that method. And then in terms of relief, it's categorized as low, medium, or high based on the 3D structure or the rugosity of that reef. And so in this case, you see a reef relief that has 20 to 60% of the sea. So we consider that about a high relief. And then contrasting to that, this is a low relief scene. So you can see very little reef complexity in here, just a few soft corals and things like that. And so and not very much rocky structure. So we don't get a high rugosity on this one. So this one's classified as low. Okay, so also for the data analysis, we take the relative read abundance. And the relative read abundance is basically the proportion of the sequences from that particular sample that are attributed to a particular species. And I'll explain that more when we get onto the graph so that you can see that. But it's not an actual direct correlation to the abundance of that particular species, because that's really hard to get at. Because it could be one fish or it could be 20 fish that contribute to the abundance in that sample. And that's nothing, I mean, people are working on that, but there's very little progress for standardizing that right now. Okay, and then we did analysis based on presence or absence of the species. And then we looked at species richness at each site. So what was that level of species richness? And then we did a generalized linear mixed model to try to understand the factors at play here. And then we also did a canonical correspondence analysis, or CPA, to try to look at whether depth was a factor, reef relief, complexity, and so on was a factor in driving the richness of these different sites. Okay, so now on to our results. So we sequenced 184 samples from 55 different sites, and we sequenced, or from these sites, we sequenced reef fish at 90% or greater identity. And we detected 48 different taxa. And we had 39 unique species, so we were able to identify 39 of them to actual species level, 44 genera, 35 families, and 2 classes. So when we look first at the surface versus the bottom water samples, 17 of our fish taxa were shared between both of those 2 samples across the sites. And if you look at this map here, you can see surface samples are in diamonds, bottom water is in a circle, and where we had both samples, we had a square. As you know, in field sampling, it doesn't always go perfect, and you don't get both at every site. But where we did, we saw 17 fish that were shared. And then the surface had 11 unique fish taxa, and not surprisingly, the bottom water samples on the reefs had 20 unique fish taxa. So greater diversity on the bottom samples. So bottom samples had more unique taxa. And this has been found when we think about fish abundance and species richness, it's greater on deeper reefs. And that is basically what we saw across the board with it. Okay, so I know this is kind of hard to look at in terms of the table of the results. So I'll just highlight a few of the species for you. So, and first, this is the surface. These are the surface samples that we got. So you can kind of see if you're curious about the different fishes that we recovered. And they're kind of stratified, hopefully kind of by where they are placed in the water column as well. But we get things like, some of the most abundant species were big eye scad, Atlantic moonfish, rough scad, which are all not surprising in the open ocean pelagic zone. The Bennett's flying fish, the goby was a bit of a surprise to us. Spot croaker, Gulf king croaker, Atlantic croaker, bullet tuna, least puffer, and sea robin. So you can see we did end up with some more benthic species in the surface samples, and I'll explain that in just a little bit, or some potential explanations for that in just a little bit. And of course, we had lionfish in every sample almost. Lionfish were everywhere in our data. Okay, and then some of the benthic samples. Obviously, the species richness is greater in the bottom water samples. Kind of at the top, really striking was a wolffish, and we'll talk about that in just a little bit. Rough silverside, striped codlet, longfin yellowtail, sandbar shark, Gulf menhaden, round eye herring, snapper, Red snapper are incredibly abundant. We'll talk about those in just a minute. We did get more lionfish, some skates, batfishes, bluefishes, croakers, drums, pinfish, kind of some of the usual suspects that you would suspect, and then others that are not probably seen on cameras as much, like the purple mouth moray and the horned lanternfish and some things like that. Okay, so in looking at our relative reed abundance, these graphs that you see here, on the y-axis we have percentage of the reed abundance. So we go from 0 to 1 basically, and the percentage of fish at that particular site is represented by like the abundance of those species in that sample. So it doesn't mean like we have a fine red snapper, since that was the most abundant DNA in that particular sample. And so what we see by and large across the board, so these are our surface samples and these are our bottom water samples. What we see with red snapper was the most detected species in both the surface samples and the bottom water samples. And that's probably not surprising to you if you know anything about reefs in the Gulf of Mexico. There are lots of red snapper. So we had more than 50 detections across 38 sites from both surface and bottom. And that represented 31, almost 32% of our reeds. And it was detected at a greater frequency in the western Gulf of Mexico. And this is not also surprising because there's a greater fishing pressure in the eastern Gulf of Mexico. And at those sites, those are at more shallower water sites. And so we tend to have deeper water sites in the western Gulf. And that's where we tend to get more red snapper. And then additionally, in the western Gulf, we have the Flower Garden Banks National Marine Sanctuary, which is protected habitat. And so ideally, that's potentially doing its job. And having more fish in those sites. Okay, the second most detected species throughout our study was the red lionfish. And of course, they are hybridizing in the Gulf of Mexico. So we're calling it Pterois volitans, but that also encompasses Pterois miles because they do hybridize. So we had 29 detections across 28 sites, and that represented almost 20% of the reefs. And as I'm sure you know, this is an incredible invasive in the Gulf. And they are heavily distributed throughout the Gulf of Mexico. And we've seen with lionfish that they don't appear to have any preference for reef relief. They're just on a reef. If they're structure, they are there, even into freshwater habitats. And so we didn't see a lot of correlation with where they were. They just seem to be everywhere. And this, I wanted to mention too, on these figures, the colors are not correlated. So like, the species color doesn't translate to the bottom because the species are not the same. But I've highlighted like the lionfish are the brown and this one and this one are kind of the lighter green. Okay, and then other species that were more abundant are some of the most detected species. So the striped codlet was another at 5.2% of the reeds. Glass eye was 2.7. The bay anchovy was 2.5. And then everything else comprised less than 1% of the taxa. So by and large, the majority of the species were the reeds were red snapper and lionfish. But we have a whole long list of more than 40 other species that comprise small components of that. And so what that tells us then is that those individuals are there. They're just not there in incredible numbers. Like we're seeing with the snapper. Okay, and on our reefs, the species richness range from 1 to 7, which is low. We think that's really low when we're looking at some of these reefs. And part of that, so even though we got a long list of like who you might expect, we didn't see a lot of richness at each site. And there's a variety of reasons for that, which we're still exploring. And one of those is that we're comparing the data right now with camera data. So we want to see what the richness is with the camera data. And they actually think that some of these sites, it's relatively low. And part of that is because when they put the camera down, we think we're on the reef, right? So the reef here, and put the camera here, you're not actually getting perhaps a full picture of the reef. So we need some optimization with that method, but that's just sort of a hazard of using cameras and being on the reef or putting the CCD where you think it is on the reef and so on. So we're working on optimizing this in the next few years, but it was relatively low at each particular site. Okay, and then with the GLM, we saw no significant relationship between species richness and environmental variables. So those environmental variables that we looked at, like surface versus bottom zone, different complexity and biotic zone. So this didn't really show us very much when we did this analysis. And I think the reason for that is because species richness is low, right? And if we had greater stratification of richness, we probably would have seen some significant results here. And so stay tuned for hopefully the next few years of data, and we might get some better data for this. But you'll see that sample zone and sample depth are like approaching that edge of significance. So we're very close there. And so we see different species lists, we see different communities in those zones. And so, richness is just a count, like we just say 13 and 13, it doesn't compare who's there, right? So that can be one reason for that. So to look into that further, then we did a CCA. And this helps us to look at the different factors and correlate each specific species as to why they're there in terms of those particular drivers. And so I'll just point out a few for you. So one of those is flatfish. And this is driven by depth and complexity, which isn't surprising, right? Because these fish live on the bottom. And so they are driven by low depth, like, on the bottom, basically. Another example would be Atelius, which are a snapper, which are heavily driven by deeper reefs. So we found those on deeper reefs, which if we think about the ecology of these fish, we understand that. The plainhead filefish was driven by relief and depth. So the deeper the reef and the greater the relief, we saw the plainhead filefish more often. The silvery lightfish, we saw the silvery lightfish, the glass eye snapper, the wolffish. Those three were kind of all in the middle, driven by complexity and depth. So the deeper the reef and the more complex the reef, we saw more of those. The Atlantic moonfish is an outlier on this particular graph. So it doesn't really seem to follow any pattern in what we know about moonfish. They're up in the surface water column. So I think it's probably due to the low detections and it's an outlier, but it is still nonetheless hanging out over here with reef relief. We did see the horned lanternfish, which is kind of an oddball, right? It's not exactly a reef fish, but a lot of these reefs were sampled at the edge of the continental shelf, where a lot of these fishes are probably migrating up during the period in the evening or in the night. And so the sampling was first thing in the morning, so at dawn. So it is quite possible that we did see some of these fishes. Okay, and then red snapper right smack in the middle, same with lionfish. And what we consider others, every other fish that was on this list. So red snapper and lionfish seem to just be kind of a combination of all of these factors. Okay, so overall, red snapper were the most detected species. They were previously listed as vulnerable, although they've been moved off of that list, and they were our most abundant species. There is ongoing debate, though, with management plan about where the species should be, if it should be listed as vulnerable or if it's doing just fine. If you've read any red snapper literature, you know that there's a hot debate about the status of it. And so eDNA is great for continued assessment of this over the next few years on these reefs to kind of see how abundances across these different sites are changing in correlation with camera data. Okay, and then lionfish was the second most detected species, and little is known about the abundance trends. It's thought right now that lionfish are starting to kind of plateau in the Gulf of Mexico, but for a long time they were just continuing and up and up and up. And you guys have probably heard about all the lionfish like rodeos and things like that. People pull 20,000 individuals out of like Pensacola, for instance, in one day. So little is known about that abundance and how they use their habitat and their wide distribution. So this is a good tool to help continue to monitor for that. And then there was a recent study that was done by Crollo et al. Out of the Pillar Lab looking at artificial reefs. And so they filtered several liters of water to look at the fish. And we actually had a higher number of taxa than they did. So what that tells us is that with our precipitation method, which we only sampled 45 milliliters of water compared to 2 liters of water, we did relatively well. So they got like 32 taxa, we ended up with 44 taxa. So that's not bad in terms of being able to match some of these sequences to who they are in the database. So, I mean, overall for this, we successfully used eDNA metabarcoding to detect fishes in the Gulf, but we have a lot of room for improvement in terms of study design and hopefully improving the actual information that we garner from this. And as I mentioned, we did get that low species richness, and that could be due to patchiness of DNA in the sampling volume. So just because we sampled such a low volume, you only get a few species in that particular volume, and that's definitely plausible for that. And that diversity of taxa has directly influenced that. Like data show us the more water you sample, the more species you get. And that makes sense. Like same thing with a conventional method. The longer you seine, the more species that you pick up. So why should we stick with this? Why do we stick with the preservation method? Well, as I mentioned in the beginning, this is a method that can be transferred across several different vessels, several different scientists or staff, and have a lower risk of contamination. So for right now, NOAA is really interested in doing that and using this method as a potential option. And then for this year, for 2022, we actually kind of changed up our plan and we actually decided to take more samples. So we got some preliminary data back and we saw we kind of need more water. So we took more samples at every site too to hopefully increase that. Okay, and then another thing about the low species richness is that our database for the Gulf of Mexico is incredibly lacking for 12X. So more than 60% of the species are missing from the database. So like red snapper, for instance, isn't online. It isn't in the GenBank for the 12X gene at the particular place where these primers are meant to amplify. So we've added to that database and we're working on building that. We've added 200 species right now. But like families of fishes that you know really well are completely lacking from the database. There are no butterfly fishes, many snapper that are missing, many bat fishes, many groupers. And so you might be saying like, why didn't you get a gag grouper? Why didn't you get some of these common species? They're not in the database. So we had more than 50% of our reefs aren't assigned. So more than 50% of our species, we can't say who they are because there's no voucher in the database. The good news is once we build that database, we can go back, run our pipeline again, and get assignments of the species. And with our new assignments, we're seeing greater resolution for this. So it seems to be working, but it is a work in progress. Okay, so this is a powerful first look at using eDNA to help assess natural reef fish communities in the Gulf. It's incredibly complementary when it's used in combination with conventional sampling, such as video surveys and so on. And it's the foundation for the rest of the NOAA Restore G. Fisher project, which I'm a part of, and that we're continuing to sample. So we're doing conventional camera data and then also the eDNA methods for that. Okay, and I think I still have a little bit of time, so I'm going to tell you the second story. So the second story today is about the least turn, and this is done with Audubon in the Audubon Delta, where we collected bird poop and started to look at diet content of these birds. Okay, so we use the same kind of foundation for it. So we use DNA metabarcoding. So we're not calling this eDNA because it's not like DNA from the habitat, but we ideally just walk up to bird poop and put it into the tube and then use that same DNA metabarcoding method. So it's glamorous work, right? Collecting that. So this is the least turn. You probably see this bird quite a bit around this habitat, so you're probably familiar with it. It has a wide distribution, and we wanted to really look at the diet of this bird. We know that a lot of diet for birds based on conventional methods. So like you see drop fish, like what fish you actually see in the bird's mouth, and then do they drop that fish or do they eat that fish? And that's kind of the data that are used to kind of to observe that. Or you might observe stomach contents, and that requires sacrifice of the animal, right, to get into the stomach. And so what we wanted to do was do DNA metabarcoding of fecal samples, and this helped to provide a non-invasive, right, because bird is getting the poop, potentially accurate, more accurate, and cost-effective solution for potentially looking at the diet of least turns. So we collected over 3 different years, and unfortunately, the birds don't go to the same places every year, so there's different colonies every year in different locations. So when you look here, we have zone 1, zone 2, and zone 3, and they were collected over different years, but we still stratified them based on these zones to be able to understand the differences in these birds and kind of the same birds that come back to these colonies, which are sampled by the Delta Audubon every single year. They go into these colonies, they monitor these birds, and they kind of have a feel for the colonies that are returning and where the colonies are shifting. So the exact sites are not the same every year, but the zones kind of maintain that frequency. So I'm not going to go through the methods because they're basically the same for everything that we did for the fish metabarcoding, but we're still here looking for fish. So we use the same primers, same bioinformatics, same pipeline, all of that. We just use bird poop to do that. So what we found when we look across the years is some great difference. So we see Anchoa mitchellii Anchoa mitchellii is, a really common fish and a fish that least terns eat quite a bit. They increase by year, but Anchoa hepsidis is notoriously lower for 2019. So we see that particular fish decrease quite a bit. So here you see Anchoa mitchellii in the red, and you do see it increase, but you can see here Anchoa hepsidis in kind of this brown yellow color. It was kind of the same for 2017 and 18, but then a drastic decrease in 2019. And we see a contrast in lionfish decrease by year. So we had a lot of lionfish, which are represented by the green bar in the first year, and then less of that in the second and third year. And we don't actually think these birds are eating big lionfish, right? We don't actually think they're pulling out of massive lionfish. We either think it's like secondary predation. So other fish are eating larval lionfish, and then we're getting that detection through that. Or these birds are actually getting small, very, very small larval lionfish and eating them as well. Okay, and then we also see a change. Oh, sorry, I forgot to animate as we go. There's a lionfish. And then we see membranous martinica. We see a much higher abundance of the relative reed abundance in 2019. So very small in 2017 and 2018, and then a greater increase in the prey for 2019. And then also we see Fundulus grandis and Brevirida tyrannus. They're mostly are present in only the 2019 samples. So we see a shift in diet from year to year when we look at these different colonies from those different birds. Now, when we look at them based on zone, so remember I told you like they were differences in the zones and they were but all collected from different years, but same kind of places on those maps. Anchoa mitchellii was the most abundant fish, and you can see that here in red for the 3 zones. So when we looked first at years, now we'll look at those zones. If you remember zone 1, 2, 3, that's the most abundant fish that we see. And then we see pterois was mostly present in zones 1 and 2. So that third zone was really high, kind of up on almost a freshwater area on the river there, and we don't see a lot of lionfish in those fecal samples from those birds. And then also based on zone, I want to mention Fundulus grandis and Medina. They were mostly recovered for zone 3 only. Did I highlight them? I guess not. But those fishes, so you can see here in purple and in in blue, those fishes were the most abundant in zone 3, not really in zone 1 or 2. So some definite differences based on zones and years for collection of that. And then one other thing that I want to tell you about that we looked at, which ended up being really cool data, were chicks versus adults. So we actually had chick fecal samples and adult fecal samples. And what's interesting is that for the adults, so you see the adult bar on the left and then the chick bar on the right. So Anchoa mitchellii represents more than 60% of the adult diet when we look at those fecal samples, but only about 30% of the diet for the chicks. So the adults are definitely eating that fish, but less for the chicks. And then interestingly, membranous martinica was the most abundant prey item for chicks compared to adults. And if you think about these fish, oh, and then one more, let me show you. Brevirida tyrannus was frequently recorded with drop fish data. So in conventional methods, the scientists go out and they say, hey, we see this bird, pick up this fish, and then they drop it. So we assume that they're probably going to eat that fish. But actually what we see is that the Brevirida tyrannus, which is a really wide body fish, isn't being eaten by the chicks at all. And instead, these skinny, narrow body fishes are being eaten by the chicks. And if you think about that, if you're a little chick, it's really hard to get a broad body fish down. It's really easy to get a skinny fish down. And so that makes sense. But conventional data with drop fish data, we assume they're eating those bigger broad body fish. And the Metabarcoding data tells us they're not. They're not in their diets. Okay, so how does it compare overall to drop fish data? Well, really interestingly, we have more fish detected with drop fish data than the different species we have in our limit. And so what that tells us, there are a lot of fish that we think that they're eating, but they're not actually eating. So with the drop fish data, we had 32 fish. So we saw 32 fish that were ID'd like by, relatively good scientists in terms of fish ID. And then Metabarcoding identified only 21 of those taxa. And out of the 21 taxa, only 6 were reported for the drop fish data. So the rest of the taxa that we collected with PGB Metabarcoding were never even recorded with drop fish data. Now, some of these are kind of a simple answer, like, with dropfish, we said it was Gambusia affinis, but it's actually Holbrooki, so it's actually just a closely related species. So here are 3 species that we got with both Anchoa hepsidus, Anchoa mitchellii, and Herringona jaguana. And then species that we found only with the dropfish data, but did not recover with the DNA data. So we didn't cover mullets, we didn't recover the Breviruta patronus, we actually recovered Breviruta tyrannus, right? So the conspecific, but that's like probably a mis-ID from the dropfish data. And then of course with dropfish, we did not recover silver size and we didn't recover lionfish, but we did with the environmental DNA. So in summary then, for the leaf turn metabarcoding, we sampled 21 fish from 83 fecal samples using DNA metabarcoding, and Anchoa mitchellii maintained consistently the main fish fry in adults, but not chicks, right? So we always think that the chicks are eating the same thing as the adults based on our dropfish data, but they were not. And we observed differences in the composition of taxa based on the relative reed abundance, based on the year and the zone. So we see some shifts in what these birds might be eating based on where they are, and then also based on the year. And then the dropfish data reported more species in comparison to metabarcoding, but both data sets are complementary. So in combination, this gives us a really powerful look at what these fish are actually consuming, or sorry, what those birds are actually consuming and eating. So before I end, I'll just have some acknowledgments because of course this one was not on the final, and I didn't do most of the work that my students did or postdoc did. So I'd like to thank my postdoc, Dr. Vic Bogante, who is now an assistant professor. A lot of the fish work for the first project was done by Samantha P. Angster, who's now working with NOAA. Other members of the Janosik lab, the NOAA Pisces scientists and crew, of course, the funding for this project from the NOAA Restore G. Fisher project and the Hal Marcus College of Science and Engineering. If you want to learn more about the Janosik lab, you can check out our website here. Also you can follow me on Twitter I'll see how long I'll be there.